muscle, actin and myosin myofilaments lie side.
This is as loud
In a
as I can get
relaxed
it to.
muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins are pulled toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes.
What I want you to pay attention to is watching what happens in my first. What's moving?
Myosin myofilaments
when the muscle's
interact.
contracting.
The
What
actins
molecule
are
is
pulled
moving?
toward the center
Purple or
of each
green?
myosin myofilament. As a result, the
They're both kind of moving
sarcomeres
in their own
shorten.
little way.
In the fully contracted
the
muscle,
actin, that green thin filament, it's the one getting closer and closer together. The myosin head is, grabbing onto it with those little hands and pulling that green actin closer and closer together towards each other. So that eventually those two green ends are going to go farther and they're going to get closer and closer together. That's what happens. Oh, I can do this. That's what happens when a muscle shortens. When you shorten a muscle, this is all you're doing is you're making that myosin and actin go past each other. Each one of these units, this whole part where we go from one, they call it a Z line, it's the end, to the other one, that's a sarcomere. As I tried to draw one down here, it's not as cool as that looking. That's all right. But that's just so you kind of have like a basic. That's what it looks like when a muscle contracts. Now, on the video, it leaves off right around here. I'm actually backtracking just a tad. There are two main steps that have to happen when a muscle contracts. We have to have an activation step. I don't know what slide number this is. It's approximately page 13-ish. It has to go, a muscle has to contract, or for a muscle to contract, it has to have an activation stage, which means that we have to have an action potential. Why does action potential sound familiar? We didn't learn about it. It's amazing. And so we have to have some type of electricity happening. And this happens at the neuromuscular junction. What two things do you think come together at the neuromuscular junction? A neuron and a muscle. Amazing. And so we're going to generate an action potential there. After we generate an action potential at the neuromuscular junction, we then have to have step 2. It's called the excitation-contraction coupling. We have to excite something, there'll be another action potential, and then the muscle will fully contract. So it's kind of like something's happened at the neuromuscular junction, something's happened eventually at the muscle level to actually cause it to contract. Now, the biggest thing, and I made it red for a reason, and I even color-coded my thing red, we'll be paying attention to calcium ions and where they're at, because we need calcium. It's not just, calcium's good for the bones. Calcium is needed for muscles to contract as well. If you're still trying to figure out where we're at, I think it's right. If I can, where's mine? Just doesn't want to work today. There we go. Big old picture that has this. These are all the steps of phase 1, which is the activation, and phase 2 is which is the excitation-contraction coupling. So phase 1, that's all activation. Phase 2, excitation-contraction. The orange part and the pink part. We're going to go through these two phases. So we're going to go through everything in phase 1, the activation, all is at that neuromuscular junction. Phase 2 is all what's happening in the muscle itself to contract. So we're going to kind of go through all the steps that eventually get to a muscle actually contracting. So starting with phase 1, what's happening at the neuromuscular junction? 1, we have axons. If you remember what a neuron looks like, it's got that cell, it's got the cell body, big long axon. What's at the end of the axon? The little terminals, because that means the end. At the axons of a somatic motor neuron. I italicized somatic for a reason. What does the somatic nervous system control? The muscles, the skeletal muscles specifically, which is what we're talking about contracting today. So we have axons of a somatic motor neuron, travels from the central nervous system via nerves to that skeletal muscle. So we've got information going from spinal cord to the skeletal muscle. Each axon at the end forms several branches. That's all of our little axon terminals. And these axon terminals, the little ends here, and they usually even have little bulbs on the end, that's what's actually going to interact with muscles. The axon terminal, which is by way over on the left-hand side, that top left thing that I labeled axon, that's one of these, the little axon terminals, the little bulb at the end. And so each axon ending or each axon terminal forms a neuromuscular junction with one muscle fiber. So we could have one little muscle fiber right there. There you go. Axon terminal, muscle fiber, neuromuscular junction. So that's what a neuromuscular junction is. It's the end of the axon. Here's my end of the axon. Little bulb, my little axon terminal, interacting with a muscle. This is my muscle. Kind of everything below here is all going to be muscle. So my neuromuscular junction, yes, they're usually situated somewhere along the length of a muscle fiber. Muscle fibers, it's a fancy way of saying a muscle cell, are very, very long. They run the entire length of a muscle. These neuromuscular junctions can be anywhere along the along that muscle fiber. It doesn't have to be at the end or right in the middle. It can be situated, somewhere along there. The axon terminal and the muscle fiber are separated by a gel-filled space. I don't know what, I'm like running out of colors. The gel-filled space called the synaptic cleft. This is the little synaptic cleft region. No, I want green. Sure. No, I don't want green. So that space in there, it's not actually touching each other. Inside the axon terminal, I have little round structures. These are called vesicles. Vesicles are just like taxis. They just transport things around. In particular, the thing that is getting transported in this vesicle is a neurotransmitter. And I made it green, so it's known as acetylcholine, abbreviated ACH. It's easier, right? So inside that axon terminal, there is a neurotransmitter that is carrying, or there's vesicles that are carrying a neurotransmitter called acetylcholine. On the muscle. Now if you notice, right underneath that terminal, the muscle's not a nice straight line anymore. It has all of these folds. We'll just call them junctional folds. It's at the junction where two things meet. Along these folds, we have acetylcholine receptors. That's what my little purple circular things are. These are my acetylcholine receptors. So the goal, hopefully, is to get the acetylcholine that is up in these vesicles down to the acetylcholine receptors, and they'll do something. Now, picture-wise, if you're like, I can't see anything, she's right. I'm trying to draw this. Just not all of them. So you've got all these vesicles with acetylcholine. I tried to color coordinate it with a little green acetylcholine. Along these folds, you've got the protein here. These are your acetylcholine receptors. That the goal, hopefully, is to get these little green acetylcholine molecules to attach to the acetylcholine receptors. So our hope, it's kind of like the quick summary of what's going to happen here, is we are going to have a nerve impulse, action potential, arriving at the axon terminal. And so I'm like, they're showing it right here. It's kind of our first step. It's in my little pink arrow. There's an action potential. This is the nervous system. It's the end of an axon. I'm like, we've already talked. We just got done testing on it. I'm like, action potentials are created in the nervous system. Action potentials travel along those axons. And as they travel along the axons, they may end up at the axon terminal. They should. And in this case, it will end up here. When that nerve impulse, that action potential, should have made it another color, arrives at the axon terminal, the acetylcholine that's in here is going to get released into the synaptic cleft. The The acetylcholine is going to go through the cleft, go find itself a receptor, bind to that on the sarcolemma, and then hopefully we're going to create another action potential. So we start with an action potential, we're going to generate an action potential. We can't just go, here's an action potential and it travels all the way across. There is a gap here. And so we can't have one action potential just naturally going across. We end with an action potential, we have to generate an action potential. Now, a sarcolemma, just in case you're like, I don't even know what that is. It goes back to prefixes. Sarco means muscle. It's one of the prefixes. There's several prefixes that all mean muscle. Lemma means a membrane. So a sarcolemma? Muscle membrane. It's that plasma membrane, phospholipid plasma membrane bilayer of a muscle cell. It's that exact same thing you find on the outside of every single cell. Well, we're talking a cell here. Except instead of calling it the plasma membrane of a muscle cell, we just give it its own name, the muscle membrane, and give it its own cool name. But we're going to go into a little bit more detailed steps of, how we start with an action potential, how we end with an action potential. So I tried to break this down. These are all the same pictures that are in your book as well. This is where I'll probably draw a few more things on here. We're going to start with that action potential that arrives at the axon. It goes to the axon terminal of a motor neuron. Again, what's the difference between a motor neuron? Or what is a motor neuron? Different than a sensory neuron. Yeah, it's information leaving the central nervous system to go do something, specifically, some type of motion in this case. So we are going to start with an action potential coming from the nervous system. As soon as that action potential gets into this axon terminal, that little end way down there, when it gets in there, voltage-gated calcium channels are going to open. Now, a voltage-gated calcium channels, if you remember, there's different types of channels. A voltage-gated one opens if there's a change in electricity. Well, if we have an action potential coming in, we've changed electricity. And so these voltage-gated calcium channels are going to open. And calcium, which we find a lot of on the outside, is going to go into our axon terminal. So it's going to want to go into the axon terminal. It's going down its electrochemical gradient, meaning it's very positive on the outside. It wants to go on the inside. Why do we care about calcium at this point? Because once that calcium goes in here, once I have calcium molecules getting into the axon terminal, it makes these acetylcholine vesicles fuse to the outside and release their acetylcholine. So now I have acetylcholine going from my axon terminal into my synaptic cleft. So I'll have lots of acetylcholine all going into my synaptic cleft. Calcium made that, made those vesicles. Their vesicles are like, we don't like all this calcium. Let's go fuse. Get rid of our acetylcholine. Where's the acetylcholine going to want to go? To the receptors. So it's going to go across the synaptic cleft, make its little way until it finds one of those acetylcholine receptors, and it will bind to it. So we're at this point. Acetylcholine, little green molecules, going across that synaptic cleft, and they're going to bind at those acetylcholine receptors. Why this matters is because once those acetylcholine receptors are full, meaning you've got enough acetylcholine attached to it, they went down there and attached to it, that receptor changes and it opens the channel up. And two things are going to go across to the channel. Sodium is going to go into the muscle fiber, and potassium is going to leave. Why does this sound familiar? Sodium going in. The sodium potassium pump actually reverses it, kicks it out. This is what's happening at that club axon. The sodium will rush in. Eventually the potassium will all leave. This naturally happens when the acetylcholine receptors are full. It opens up. Sodium is going to rush into that muscle cell, and the potassium is going to leave. Sodium, there's lots of it on the outside, so it wants to go from where there's not as much. And same thing with potassium. There's lots on the inside, so it wants to leave as much as possible. When sodium is leaving and sodium's going in and potassium leaving, it changes the ion concentration. It has not created a full action potential yet. It has, however, generated a small change in electricity, which we call the end plate potential. So it's not an action potential yet, but it generates some electrical change, and we call it an end plate potential. Reason why we call it end plate, another name for the sarcolemma, once it has all the folds, the sarcolemma part where it has all the folds is also called the motor end plate. They call it an end plate potential. We're generating some type of electrical difference here along all these folds. So it's called the end plate potential. So we started with an action potential. We almost generated another one. We're getting closer. Once we've generated this slight difference in electricity, this end plate potential, we kind of want to be done there. We don't want to keep generating it. There is an enzyme known as acetylcholinesterase. Very long word. Most enzymes end in an ace, and then they just, go back and tell you what that enzyme works on. So acetylcholinesterase is an enzyme that breaks down or does something with acetylcholine. It breaks acetylcholine down. So it takes that circular acetylcholine, here's that acetylcholinesterase, and it takes it and breaks it down. So that receptor will close back up again. We've created that end plate potential. Let's get rid of the acetylcholine, and we're done with it. We did our job. We created a little bit of not action potential, but we've created a little bit of electrical difference there. So far, we had that nerve impulse, action potential, arrived at the axon terminal. Acetylcholine goes in the synaptic cleft, goes across, finds a receptor along the sarcolemma, and we are starting to create an action potential. We've created the end plate potential, but we can't call it an action potential yet. Now, I kind of briefly said it, I'm like, yes, but we don't want that acetylcholine there forever, so the enzyme comes in and breaks down acetylcholine, the acetylcholinesterase. It prevents muscle fiber contraction in case there is no stimulus. So you don't want to I'm going to lift my book up, and then all of a sudden your arm just never moves again, and you're like, well, I want it to not be moving anymore after I picked the book up. And so I'm like, if you're not sending any electrical information to your muscle's not going to contract. So it does its job, acetylcholine does its job, and then it goes away and gets broken back down again. Let's see how well this video. I don't know how loud you're going to hear it.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
I'm going to start all over again. Now, how well can you guys hear it? I know it's not going to be loud. Can you hear it, though? That's nothing. I'm going to get this. Yes, you could read it. I don't know. It's kind of small from the far back. I just can't make it any bigger. The website only, it doesn't get it bigger. Even if I make the screen bigger, it stays the exact same size. But you kind of saw what happened. You know, that action potential went in, calcium channels opened, acetylcholine got released, it opened the channels, sodium rushes in, create a slight change in electricity, potassium will leave, and we have our motor end plate potential. Now, we still have to generate a full action potential. So our sarcolemma, our muscle membrane, becomes polarized with this end plate potential. I'm just going to label this sarcolemma. This is the muscle membrane. It becomes polarized or charged. We're going to generate an action potential. And it's caused by changes in electrical charges. That's anything with electricity. And it happens in 3 steps. The first step we've already done. We've already created the end plate potential. So local depolarization, that just happens here when those acetylcholine receptors opened. That's all we've done so far. So the acetylcholine binding, it opened those gated channels. Sodium goes into the cell, potassium goes out. Since more sodium goes in, it becomes less negative because it's positively charged, sodium is. And it creates a local depolarization, or that end plate potential. So we've done that already. This is what's happening. Should look familiar. Depolarization, repolarization, hyperpolarization. Same thing that happens, but it's just happening in a very small scale. The end plate potential. But then we can take that end plate potential and depolarize other parts of the actual cell itself, other parts of the muscle membrane. So that end plate potential, the fact we've generated like a little bit of electrical current here on the folds, it's going to spread to adjacent membrane areas. So it's going to spread. I should use different than black. That end plate potential, that electrical information is going to travel along the sarcolemma. And it's going to cause voltage-gated sodium channels, which that's what it is. There's a sodium channel to open. And where do you think sodium wants to go if a sodium channel open? In or out? Think of a club. It wants to go in, and so we're going to end up having sodium going into the muscle, creating more positive charge. As long as enough sodium goes in, you'll meet threshold. You will now create that action potential. And so again, we are going to create that full action potential. It's not just a little localized at the end plate, a little bit of electrical difference. We've now created a full depolarization. We've created a full action potential. And as long as I hit threshold and enough sodium went in, my muscle will contract. Kind of we're at the all or nothing part right here. Now I'm like, once I've hit threshold, I've generated a full action potential right there. A depolarization has happened. My muscle will contract. Other things are going to be happening still, of course, to make the muscle contract, but a muscle will contract. And then just like any time you create an action potential with depolarization, I was going to say we'll have to have repolarization. Before I get to that one, I have one more slide. When this sodium channel opens, it's just like what happened at Club Axon. A couple of those sodium girls snuck in, and then they told all their friends how to do it, and then another sodium channel opens up, and more sodium go in, and we create another action potential this way, and it will continue to travel along that muscle fiber, causing, action potentials to travel along the actual muscle membrane. Eventually, we have to have repolarization. If we have depolarization, things have to kind of reset themselves. The sodium channels will close. They will close. We will have potassium channels open. I didn't draw any more in. We will have a potassium channel open in, and we will have some potassium leaving, to try to get our charge back to normal. Now, at this point, a fiber cannot be stimulated anymore. It's what's known as a refractory period. When you can't have another stimulus happen, you can't have another muscle contraction happen, you haven't even had this one happen yet. And eventually that sodium-potassium pump will make sodium leave and make potassium go back in eventually. The big thing I care about is we've created an action potential on the muscle membrane, and it will create further action potentials along the line. This, up until about, where I drew that K off to the side, this so far is all phase 1. This is all phase 1. This is all that activation phase. We started with an action potential in our nervous system. We end with an action potential on our muscle membrane. But have we actually made a muscle contract yet? No. Just got some electricity going on. So we had that end plate potential, local depolarization. I went from one Windows program to another. All the little squares are supposed to be negative signs. Not that it matters. The big thing happened, depolarization, sodium goes in, repolarization, potassium will leave. We've created a full action potential along that sarcolemma, along that muscle membrane. As you can see, a sarcolemma, muscle membrane, still that phospholipid bilayer, muscle cell membrane. I've created the depolarization, I've created the repolarization. Should look familiar. But we've gotten to the end of phase 1. We started with action potential, we end with an action potential. Now the question is, how do we take this action potential along that sarcolemma and actually make a muscle contract? Make that sarcomere shorten. So the excitation-contraction coupling is taking that action potential that we've created, transmitted along the sarcolemma, along that muscle cell membrane, and eventually lead it to a sliding of the myofilaments. That's the actin, the myosin. Eventually we'll get the actin and myosin to move past each other and shorten. Now the action potential that we've created, it's very, very fast, it's very brief. It will go away, the electricity will go away before the muscle actually contracts. The biggest thing that's going to happen after we've generated this action potential, when we get into our phase 2 here, is I'm going to release calcium. Again, I made it in red. It's important. We are going to have movement of calcium. Calcium is very important in contraction. Now, my little note. There is something known as a latent period. It's different than a refractory period. Refractory is things haven't reset themselves yet. You can't have another stimulus happen. A latent period is things are happening, but a muscle hasn't contracted. So an action potential was generated, but that doesn't mean instantaneously there's going to be a muscle contraction. You're going to just see that muscle contract. There's things that have to happen. There are things that have to move before you actually see a muscle contract. So that latent period is just this time where stuff's happening that you don't visually see before a muscle actually shortens. So, getting all the way to the muscle shortening, this action potential is going to travel along until it gets to what's known as a T-tubule. First, can you see why it's called a T-tubule? Now here's sarcolemma, here's sarcolemma. It dips inward. It is a tube. A muscle fiber itself, a muscle cell, just does not go straight across. There are these little indents in the muscle cell itself. This is a T-tubule because it forms a T. This action potential is going to travel along here, and eventually it's going to go in one of these T-tubules. When it gets inside this T-tubules, it causes this blue structure known as the sarcoplasmic reticulum. SR, I'll write it out. The sarcoplasmic reticulum's main job is to store calcium. All my little red dots there. So it's just a little calcium storehouse. when that action potential gets inside that T-tubule, and on either side of a T-tubule, you've got a sarcoplasmic reticulum here, and you have a sarcoplasmic reticulum here, that action potential is going to make these channels open up, and calcium is going to go into the T-tubules. So now I've got calcium inside my T-tubules. They've left the sarcoplasmic reticulum. It's a big step. We've released calcium from the sarcoplasmic reticulum, and we are going to follow that calcium to see what it does. But action potential has released calcium from the sarcoplasmic reticulum. Now, one of the terms that was on the video, whether you watched it or not yet, if you haven't, watch it. It's called a triad. It's just because when you have a T-tubule in the middle and you have a sarcoplasmic reticulum on either side, that's three things. There's always one T-tubule. It's like a little T-tubule sandwich. Two sarcoplasmic reticulum with the T-tubule in the middle. Looks like a little hot dog. This is showing we've created that action potential along the sarcolemma. It's going to go inside that T-tubule, and on either side of it is that blue sarcoplasmic reticulum that has lots of little red calciums in it. It is going to release those calcium molecules, super important calcium molecules. There it is again, zoomed in. Sarcoplasmic reticulum releases calcium molecules. There's my calcium ions are released. Because it's where these calcium go next that becomes very important. Now, whether it's really too small, I might actually lift this up. Because if you're on one side of the room, I don't know if you can see the bottom of this. Now, actin, the thin filament, is a double-stranded molecule. To me, always they look like little tiny blueberries. So it's like two little strands of blueberries. I'm not talented enough artistically to draw circle strands that are twisted. So imagine that that is two strands that are twisted. I'm just not artistic enough. So these little things that are supposed to look like little blueberries, that's my actin molecule. Now, the important part about actin is the little dots, the dark dots on here, are called active sites because things are going to happen there. That's why they call them active sites. Now, what do you notice about the active sites? Can you see all of it? The full circle? Oh, they're covered up. There's a molecule known as tropomyosin. I tried to draw mine in brown. It says tropomyosin, kind of sloppy. There's tropomyosin that covers all of those active sites. If your active site is covered, is anything going to happen? No. So we're going to have to do something about that. There's also another protein called troponin that like anchors it there. It's like tropomyosin, you are not moving. You're going to cover all of those active sites so that nothing can happen. What do you think calcium's going to do? What has to move? The tropomyosin has to move. That big, long, brownish structure has to move. Calcium becomes very important in this because calcium is going to bind with that troponin protein that anchored the tropomyosin in place. When calcium binds with that troponin protein, it changes a little bit, which means it can't do its job anymore. And now, can you see the active sites? Yes. If your active sites are exposed, can stuff happen? That's very important. Now, the main reason why we've opened up these active sites, they're exposed, they are ready for myosin, the thick filament with these little heads on it. I always try to do it as, it's my arm and the head is my hand. These little heads are going to grab on to each of those little active sites. It can bind right there, and then it can move it. If myosin can't bind to an active site, nothing will ever happen. And so there's the hand. It's grabbing onto an active site, and it does what's known as a power stroke. It just goes from, almost like a straight, and then it does this. It just does this. As it does this, it bound to it and does this. The whole actin goes with it. Like it binds to it, and then it moves it, and you can shorten. You're moving actin now. You're moving those thin filaments. That's how muscle is contracting. The myosin head binds to an active site and cocks itself, or does what's known as a power stroke. Say a whole summary of all of this, just smaller pictures of everything. We're going to watch the quick little video that shows this happen. Big as I can make it.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Now, a cross bridge just means the myosin head actually attaches to that active site. It's just, they're stuck now. And so we had that calcium, the action potential coming in, calcium getting released, calcium binding to troponin. Now, I get it. Troponin and tropomyosin are two horrible words that kind of look alike and kind of sound alike. Tropomyosin covers the actin. Troponin just anchors it. Troponin is the thing that actually changes shape and moves the tropomyosin. So once that tropomyosin is gone, their troponin is changing shape. The myosin heads can bind to the active sites on the actin. There you form a cross bridge, and it can do its power stroke. That's happening in a mic, and if you have all of these heads constantly doing all of this at the same time, your entire muscle will shorten. Now, there's lots of channels that are opening and closing through all of this. So just a review of just channels that are opening and closing. One, starting way at phase 1, when that action potential or nerve impulse got here, we have calcium channels that opened up. That's the voltage-gated calcium channels that'll trigger the acetylcholine to get released. When acetylcholine binds to its receptors, it opens sodium and potassium channels, creating that end plate potential. So there are channels here, there are channels there. That's phase 1. The open, I was going to say, those voltage-gated sodium channels will eventually might cause that action potential, so you have more sodium channels opening, creating that action potential that travels along, going from phase 1 into phase 2. And as the action potential goes down the T-tubule, calcium channels open up again. So you've got calcium channels up there, you've got calcium channels here, you've got some sodium and potassium channels. I'm going to start from the acetylcholine receptors. They're going to change. Sodium channels opening here. Everything is eventually to release calcium so that muscles can contract. Now, so the role of calcium in contraction. If you have little calcium in your cells, the tropomyosin is going to block those active sites. You will not have a muscle contracting. This is what happens when your muscle is relaxed. So when you're just sitting there not doing a whole lot, your muscles are you're not contracting any of your muscles, you have very little calcium in your muscle cells. It's only when you want to move your muscles and have them contract that calcium rushes inside the cells, inside your muscle cells. The calcium will, move the tropomyosin, and then you can sit there and shorten your muscles as much as you want. So when you have high levels of calcium inside of your cells, the calcium binds to troponin, moves the tropomyosin. The myosin heads are going to bind onto the actin, onto the active sites, and your muscle will contract. Now, when you are done contracting your muscle and you're like, okay, I'm done, the calcium will eventually all get pumped right back into that sarcoplasmic reticulum. Just a little storehouse. And so calcium gets released, does its job. Calcium will eventually get pumped right back in and go back so that it can keep getting reused over and over and over again. Now, your muscle will continue to contract. The muscle, those myosin heads, will constantly keep grabbing those active sites and doing their power stroke as long as calcium is present, because that means the active sites are available, and there is ATP available. So we need two things available for your muscles to continue to contract. That cross bridge formation, that head attaching to the active site, it needs energy. You know, you have to have a high energized head to go and attach to it and do its power stroke. So that's where the ATP comes into place. So that power stroke, the myosin head pivots and pulls that actin or that thin filament towards the middle, the M line. That's what the M stands for, is middle, the middle of the sarcomere. And eventually it shortens. So you need calcium, you need ATP. Now, one of the reasons why you also need ATP is like you need that high energized head to do its power stroke, but you need ATP to like re-energize that head. So after it does its power stroke, it uses up its energy. And so it needs ATP to release the myosin head and energize it again to do another power stroke. So ATP is actually needed to detach the head and energize that head. So the cocking of the myosin head, making it have lots and lots of energy ready to do its job, you need ATP. So ATP releases the head and energizes it so that the head, with all its energy, will go ahead and attach to another active site. So this is just showing kind of over and over again, and I'm like, the cross bridge, it attaches, it does its power stroke, it had lots and lots of energy, did its power stroke. ATP will help release it and energize it again, and I'm like, it has, breaks down that energy, it's highly energized, and it's going to sit there and attach to another site and keep doing that. So it's this constant supply of ATP. And as long as you've got calcium, active sites are available. I was going to say, this just breaks it down. Not going to go through it all. You need ATP. Well, as long as you have ATP and calcium, you're good. We're not going to watch the video. Now, have you heard of rigor mortis before? What is rigor mortis? About reading that. The body, it kind of starts to contract and stiffen, and you move, and the body becomes stiff after you die. This happens in the muscle, so it has something to do with all of this and muscles contraction. So cross-bridge detachment requires ATP. So the muscles will contract and do their job, shortening the muscle, and you need ATP to relax. What happens about 3 to 4 hours-ish, depending on temperature and that kind of stuff, the muscle starts to stiffen up to about 12 hours after death. Reason why this happens: after you die, your cells start to die right away, and they start to degrade. As your cells are starting to degrade, the sarcoplasmic reticulum, which stores calcium, starts to degrade. As this breaks down, it releases calcium. If you now have calcium in your muscle cells, whether you're dead or not, what are the muscle cells going to do? They're going to contract. And I'm like, so they're going to contract because your muscles are breaking down and releasing calcium. And since you're not alive, you're not making ATP anymore. That ATP is what releases the myosin heads and energizes it. It doesn't release the myosin heads. And so your muscle contracts and doesn't uncontract. And that's why all of a sudden the muscle bodies become stiff after death. Muscles have contracted but have not relaxed. This is why I'm like, I've had people before, that have worked in nursing home, not nursing homes, but mortuaries before, and they're like, yeah, the muscle bodies will just start moving. Would freak me out if all of a sudden the leg just kicked up or whatever. But they said it happens all the time. Freaked me out. All because calcium's being released and there's no ATP getting made. All right, some review. So get out your colored note cards. Some of this is from, I think, before, like, or from the video, the intro video. So we'll see what you know. And if you don't, guess well. Of the following muscle types, which is the only one subject to conscious control? Hopefully you know this anyways. It is the skeletal. It's kind of the one we're talking about anyways. Which of the following muscular functions serves a metabolic function? It's heat generation. As muscles are contracting, that does help regulate your metabolism. Every time muscles are contracting, you're generating heat as well as a byproduct. Things move. The elastic components of muscle consist of which elements? If you guessed green, good job. Good guessing. The tendon, the epimyosin, the perimysium, and the endomyosin. These should kind of sound familiar because these sound like almost the same thing that covers the nerves. There was a epineurium, and endoneurium, and perineurium. A muscle itself is arranged very similarly to exactly how a nerve is arranged. But what do you think MYS stands for or means? Muscle. It's, you know, sarc means muscle, sarco means muscle, mys means muscle as well. And so it's the same type of coverings that cover the muscle. Which of the following components accounts for the bulk of a muscle fiber, which is a cell, volume, meaning it can comprise up to 80% of a muscle fiber or muscle cell volume? It is the myofibrils. So they're tiny, skinny little things found inside of a muscle cell. At the neuromuscular junction, this gets to today. At the neuromuscular junction, the muscle contraction initiation event is what? It is the Y, the binding of acetylcholine to the membrane receptors on the sarcolemma. The time period between an action potential initiation and the mechanical activity of a muscle fiber, meaning the muscle fiber is actually contracting, is called what? So the time period between the action potential and when a muscle fiber actually contracts. It is the latent period. What's the refractory period? Yeah, that has to do with the action potential just in itself, like when the channels are still open. It has nothing to do with, you know, between the time period one happens and something else happens. That's just channels are still open. What is calcium's function during muscle contraction? Why do we care so much about calcium? Is pink. Calcium binds to troponin, changing its shape so that tropomyosin can't do its blocking action anymore. And corpses usually exhibit rigor mortis. Why? And yes, you guys all got the orange. It's a lack of ATP, preventing the myosin head from detaching from the actin. We'll end there because it's a perfect spot to end.